So now we will welcome the last speaker of this session, Apoptosis versus Senescence. It's Professor Johannes GRILLARI, who is coming from the Institute of Biotechnology of Vienna. So, grüß Gott, Herr Professor Johannes GRILLARI. And he's a specialist in senescence, aging. It's hard to describe him because it's at the same time molecular biologist and biochemist and geneticist. And today the title of his talk is Skin Cell Senescence, a driver of skin aging and how to drive back. So we are happy to know the answer.
Merci bien, grüß Gott, bonjour. It's a great pleasure to be here for the first time in this wonderful location. Actually, I think I should sing my presentation at this opera, but I guess then you would run for coffee right now and not after my talk. As a disclosure, I'm on the one hand, I'm still an academic working at university, but I'm also co-founder of two companies, Eversight and Tamerna, and my lab is in part co-funded by Chanel Research and Technology, and that's part of what I'm trying to tell you today. So my lab for the last, and I don't dare to say that, for the last probably 20 years has been focused on understanding why we have differences in the speed of the aging process. So what drives biological aging? And I think this is a wonderful example of the difference in the speed of the aging process. These samples are normalized for social background. Both are musicians. You have Tina Turner at the image taken at the age of 68 versus Keith Richards at the age of 65, and you can clearly see, especially from a dermatological perspective, that there are differences in the speed here. And probably, and that's one of the ideas we had in the lab, maybe senescence is one driver of premature aging of this guy. Might also be the substances that run in the veins here. We don't really know. So you already heard the definition of cellular senescence. Just to recapitulate, it's been an in vitro phenomenon for a long time. You explant the cells from the human body, get them into culture, and eventually after reaching a certain number of population doublings, this number is termed the Hayflick limit after Lenny Hayflick, who first described this phenomenon. The cells enter this growth arrest, but they can do that also earlier if they are hit by a stressor, as we heard before, by oncogene, by hyper-oncogenic signaling, or by oxidative stress, for example. And I think also by there's something now termed something like a mechano-induced senescence by just having not right adherence factors on the cell culture plates. Very in vitro, but in vivo, how does it look like? And by now, we have evidence from several labs, from several different studies, that senescent cells accumulate in the human body at a lot of different sites, different tissues, different organs, and especially at sites where we see age-associated disease-dependent lesions in the brain, in the lung, in the kidney, in the bone, and we also have estimates from a couple of different studies of about 20 to 50% of senescent cells that we find in the skin of elderly. So can we now imagine that such senescent cells really are not only involved in suppressing tumors or promoting tumors per se on a cell autonomous level, but do they really have something to do then with aging? Because we expect that they would, if they are a driver of aging, then that they would somehow impact on the organismal aging process. And there have been a couple of papers out now from specifically from the Mayo Clinic, Jim Kirkland's lab and Van Dersen's lab, as well as from Buck Institute, from Judy Campisi's lab, that show that if senescent cells can be eliminated in mouse models, and these are, by now we have transgenic mouse models, and we have also mouse models where senescent cells are eliminated by so-called senolytics, drugs that specifically eliminate and drive senescent cells into apoptosis, that then we have postponing of almost all of the major age-associated diseases, including osteoporosis, lung fibrosis, osteoarthritis, atherosclerosis. We seem to rejuvenate hematopoietic stem cells. We have a reduction in the side effects of anti-tumor therapy. We saw that we have something called chemotherapy-induced senescence as well. And there's a paper, one of the early ones, that also shows beneficial effects of senescent cells. So in contrast to the other studies by Judy Campisi's lab, we see that if we would kill senescent cells during wound healing, then we delay the closure and the healing of the wound. On the other hand, we also see that if we have persistent senescence in the wounds, then we also impair differentiation of keratinocytes, and wound closure is also not working that well. So we have we need a balance. So how does that look like if we kill senescent cells in a mouse model? This is from Peter de Keyser's lab. He's now in Utrecht. This is the treated mouse at the same age as here the untreated mouse. The elimination, the senolytic drug used here is a peptide that inhibits p53 interaction with FOXO4, which is a specific interaction that happens in senescent cells. And I think this closely resembles to Tina Turner versus Keith Richards here. So why are senescent cells then bad in the sense of driving the aging process? Whatever the trigger for cells to go into senescence might be, on the one hand, and we heard in the previous talks about it, they stay within the tissue, they alter their functionality, they alter the differentiation status, they might be endogenously forming tumors later, might be. They also get killed by the immune system or by apoptosis in some instances. So they use up the replicative potential of the surrounding tissue, and this is what my lab at the moment considers as one of the important drivers in the aging processes, that they accumulate something like a pro-inflammatory phenotype, the so-called senescence-associated secretory phenotype, or SASP, which is considered one of these antagonistic pleiotropies, meaning that the senescence per se might be a tumor suppressive mechanism, while on the other hand, this pro-inflammatory phenotype, the metallomatrix proteinases that are secreted as well, which remodel the stroma, that this might be actually even pro-tumorigenic, and therefore might also drive this tumorigenesis in this opposite way as we would conceive senescence per se. So my lab started out to look for factors that are differentially expressed in early passage versus senescent cells, and we wanted to see if such factors then really can impact on the on the organism lifespan. And so after quite some time, we came up with at least two factors that we identified in such screens that actually do have an impact on the organism lifespan. Here's one, it's a DNA damage repair, splicing and ubiquitination factor called SNF by our lab, HBRP19 or PSO4 by other labs. If we overexpress that dose-dependently in flies, then we indeed increase the lifespan of these flies. The other enzyme that we identified is an ribosomal RNA methyltransferase that if we knock it down in fly, worm, or even in yeast, can expand the lifespan of these organisms. In that case, so we hit on a novel conserved pathway of organismal longevity by looking at cellular senescence. So, what did we do in terms of skin aging? So we know, and that might be questioned, but there are papers claiming that there are up to 50% of senescent cells in human geriatric skin. So we tried to model this situation by generating 3D full thickness skin equivalents where we mix senescent fibroblasts into the collagen matrix and then seed the keratinocytes on top and have them differentiate. And you will see more about this model in a talk after the break by a fantastic, actually she's still a PhD student in the last days, very close to being a postdoc in my lab, Regina Wein-Müllner. And so she will tell you a bit more about this model. But let me just say at this point that when we add senescent fibroblasts into this model, then we definitely see a reduction in the epidermal thickness that comes along. So can we now somehow, because obviously there must be a crosstalk between the fibroblasts that are inside of these, of the collagen matrix to the keratinocytes and an impact on the differentiation process of these keratinocytes. So can we somehow alleviate this not so good effect of senescent cells? And in our collaboration with Chanel Research and Technology, we screened a couple of plant extracts for their effects on senescent fibroblasts in that case. And we came up with one, Solidago alpestris, solidage, or goldenrod, is this yellow flowers that we have, an alcoholic extract of that impacts on senescence. So how did we find that? We screened a couple of different of these different plant extracts on our fibroblast senescence model, and this extract 1201 from Solidago decreased already in early passage and midterm passage cultures the amount of senescence-associated beta-gal positive cells. So we zoomed into that one and stayed with that extract, and especially looked at the effect on the SASP, on the secretory phenotype of after being treated with 1201 extract. And what we indeed see is that the pro-proliferative effect, this potentially pro-tumorigenic effect of senescent cell secretomes, is dampened by 1201. And we tested it by taking conditioned media before and after exposing them to 1201 onto pre-neoplastic cell line HACAT cells, and we see that we have lower proliferation of these cells. We also see that the chemoattraction, which is also known to be a functionality of this SASP, is dampened by exposing the cells and treating the cells with this plant extract. So what happens now if we put the plant extract onto senescent fibroblasts before we then include them into our 3D skin equivalent? And what happens is that we can beautifully restore the differentiation process of the keratinocytes. So meaning that indeed whatever is happening in terms of dampening the secretome of senescent fibroblasts is also active for a couple of days after being treated, and then transferred to the keratinocytes in the 3D skin equivalent. And obviously, because senolytic activity is of a very high interest in the field at the moment, we tested if 1201 would then also have a senolytic activity by specifically killing senescent cells in vitro, and we find yes, it does. It does so very slowly and very lowly, but significantly after exposing senescent cells for 39 days to this Solidago extract. Then we have about 30% of senolytic activity that we can observe. So it's not a huge and big senolytic that might have a huge clinical potential, but still there is something in there that where we were very happy about it. So now, what is this SASP? What do we find in there? It's pretty well known that we have a lot of pro-inflammatory cytokines like IL-6, IL-8. We have matrix remodeling factors like MMP1, M3. We also see some eicosanoids, prostaglandins in there. But we don't really know about extracellular vesicles and microRNAs. So one question that we had in my lab is, do we find extracellular vesicles and their microRNA cargo as one component of the SASP? So what are extracellular vesicles? Very hot topic at the moment. The Society of Extracellular Vesicles now tried to counteract the Babylonian language problem of the naming of these vesicles coming down to 3 names only, depending on the biogenesis. If the vesicles come from the endosomal compartment via multivesicular bodies, then they are called exosomes. If they bud from the plasma membrane, you call them ectosomes or microvesicles. And if they are blabbing from apoptotic cell membranes, then you call them apoptotic bodies, obviously, so that name did not change. So that's what we call them in the field now. And any known uptake mechanism that has been described before has also been shown in the context of taking up extracellular vesicles. Might it be phagocytosis, fusion to the membrane, endocytosis? All has been published in the context with different vesicles from different cell types being taken up by other cell types. So, since the biomarkers for the extracellular vesicles are not very clear yet, and we have proteins from the endosomal compartment that have also been found in the ectosomes, and we still rely on size as a marker for the EV species, which is not a good marker, but that's how we fractionate and purify the vesicles. If they are below 100 nanometer in diameter, we call them exosomes. If they are 100 nanometers to 1 micrometer, we call them microvesicles. For apoptotic bodies, they are usually larger, and they get annexin 5 positive by exposing phosphatidylserine to the outside of the membrane. So we consider these extracellular vesicles as messages in a bottle. So you have the double lipid membrane layer around it, and then you have some kind of a letter inside of the extracellular vesicles. And the question is, what is written on this letter? And by now we know that the letter tells the recipient cells a lot of different things in terms of growth, proliferation, angiogenesis, inflammation, immune response, epigenetic reprogramming, invasivity. It prepares the pre-metastatic niches at distant sites from the primary tumor. So there's a lot to say on these letters, and the question is what is the language the letter is written in? And by now we know that this language comes in the flavor of the amino acids, meaning proteins. On the other hand, on the letters of the genetic code, meaning nucleic acids in form of RNA and DNA, but also in terms of lipids, which are transported by extracellular vesicles. And we focused on microRNAs in that regard. So first we tried to see if extracellular vesicles would change their shape and distribution between senescent and early passage control fibroblasts. They do not. The shape is pretty similar to what you would expect from these extracellular vesicles after you purify them and look at them in transmission electron microscopy. Markers like TSG101, which is an exosomal marker, is present. And we see that indeed senescent cells, and in this case we used oxidative stress-induced senescence on fibroblasts, is the vesicle secretion goes up by between 3- and 6-fold, depending on the donor and the repetition of the experiment. With that, obviously, we have an increase of all the cargo molecules that are inside of the vesicles in terms of microRNAs. So almost all microRNAs are upregulated. If you look at the global level of the total amount of vesicles you have per cell that are secreted, but then we also find some specific changes in the composition of the single vesicles, which I don't tell you now. So since everything is up, what are the top 20 secreted microRNAs? We made a list and we performed pathway analysis, and what comes out is that most of the direct targets of these microRNAs seem to hit on the apoptosis behavior of other cells. So the logical experiment was then to take the extracellular vesicles from senescent cells versus control cells and see if we would, in that instance, decrease the apoptotic behavior of the recipient cells. So the experiment we performed was then really showing that we have a reduction in the apoptotic behavior of fibroblasts that were acutely stressed by hydrogen peroxide, which drives them into apoptosis. So higher amounts, not senescence-inducing amounts of hydrogen peroxide, but really apoptosis-inducing amounts. And with the vesicle from senescent cells, we indeed decrease the rate of apoptosis of the bystander cells. So this might be something bad, again, in terms of tumor development, where senescent cells might decrease the ability of damaged cells in the surrounding to undergo apoptosis, and therefore might allow some mutated cells to survive and eventually form a tumor here. So are now the microRNAs really transferred from the keratinocytes within a 3D experiment, a skin equivalent experiment? Are they transferred to the keratinocyte, to the epidermis? And therefore we inserted microRNA 39 from C. elegans, which is specific to the worm, doesn't exist in humans. We transfected fibroblasts with this microRNA, put them into the collagen matrix, generated 3D skin equivalents, separated epidermis from dermis, and then looked by qPCR if we would find this microRNA, this worm-specific microRNA in the epidermis, and indeed we do. So we have an indication that such a crosstalk can happen, at least in organotypic cultures. So if we have such a transfer, then what could be the outcome of this? And if you remember that senescent cells impacted on keratinocyte differentiation in our first experiments, we took the extracellular vesicles from senescent fibroblasts and put them onto keratinocytes, and what we indeed see is that the closure of scratches in keratinocyte monolayer cultures is faster by the presence of extracellular vesicles. On the contrast, if we expose these the keratinocytes for a long time to these extracellular vesicles from senescent cells, we seem to inhibit at least the formation of fully differentiated keratinocytes by seeing that involucrin as a late marker of keratinocyte differentiation is down-regulated after long-term exposure. So we again might have this double-edged sword of senescence, where in the short term it might have beneficial effects, while being chronically exposed to the SASP, you might have problems. Can we get this effect on keratinocytes down to a specific microRNA? One of the microRNA that we found differentially expressed per single vesicle is microRNA 23A3P. So we transfected the keratinocytes with this microRNA alone, and indeed we can phenocopy the effect that we saw with the full extracellular vesicle fraction from senescent cells. Can we find extracellular vesicles now in skin in vivo? For that, we teamed up with Ida Perrotta, a specialist on electron microscopy at the University of Calabria. And indeed, we find in skin cells ex vivo structures, multivesicular body structures with single structures that resemble exosomes inside of the cells. And these structures inside of the cells are positive for different markers of vesicles like TSG101, CD81, or CD63. And actually we see such structures even in the interstitium of the cells, meaning that, yeah, such vesicles are also secreted in vivo, although we don't know the directionality where these vesicles would go to. So we tried to isolate vesicles from human skin by a protocol that we set up, and indeed, yes, we can get vesicles out. But then obviously we are using a lot of enzymes to digest the skin, so it's not... We were not really satisfied at the end with the experiment here. So in that case, we teamed up with Ioneum Research in Graz, in Austria, where they developed an open flow microperfusion system where they insert capillary tubes that allow at a certain kilodalton exclusion barrier, the coming in and going out of molecules. It's large enough for extracellular vesicles to cross the barrier, go in these capillaries. So we got hold of some of the samples from probands that look like that. It's considered a minimally invasive technology, though, and but you can also run that in vitro on skin flaps that survive in vitro for a week or 10 days. So we tested if we would find microvesicles in those interstitial fluids that we collect from open flow microperfusion system, and indeed, yes, we get these vesicles. They are in the size, they are similar to what we get out of our in vitro cultures, and also in TEM they look pretty similar. Do we find the microRNAs we were interested in also in these samples? Yes. Yes, we do, and for example, microRNA 23 is one of the highest that we find in there. In these interstitial fluid-derived extracellular vesicles. So, obviously, one of the next steps is to look into young versus elderly subjects, and we are currently trying to get the ethical approval to run such a study. Summarized, I think, or I hope I could convince you that extracellular vesicles and the microRNA cargo are indeed part of the SASP, that we also can dampen this SASP by plant extracts, and that if we don't dampen that, a cargo is functionally transferred in vitro and in vivo, And we can add some messages of extracellular vesicles from senescent cells, which is that they talk about apoptosis to other cells, about differentiation and proliferation. And I think it's now also clear why vampires only bite young girls. It's clearly because they want to dilute their SASP. And with that, I would like to thank all the people in my lab for all the fantastic work, especially Lucy Teletsky-Zaniewicz and Sylvia Weilner, who performed a lot on the extracellular vesicle microRNA studies in my lab. Ingo Lemmermann, who was the genius visualizing the effects of the plant extract here, and you will hear Regina later, so I just say then welcome to you afterwards. And all my fantastic collaborators at Medical University of Vienna, Florian Gruber, who is in the audience, by the way, and Erwin Schachler, collaborators at Chanel Research and Technology, including Julie Latreille, Irina Berlin, Frédéric Morisot, Sandra Forestier, Gaël Gendronneau, and Nicolas Fuzzati. University of Lausanne, with whom we collaborated on the fly work, Ioneum research, as I laid out, and we are also in contact with Jim Kirkland and Julie Campisi for further developing the senolytic ideas that came up during our studies here. And thank you for your attention, and I'm happy to take your questions.
So thank you very much, Johannes. Cédric? Non, bah alors Jérôme Lamartine.
Well, thank you very much for this very nice presentation. I have plenty of questions that we will discuss later. Just one for beginning. When we speak about microRNA and exosome, the main question is how the message is put into the bottle. So how the microRNA are selected to be inside the microvesicle? So do you have ideas about that? And can we modify this selection of microRNAs? Perhaps, I don't know why. But.
Yeah, absolutely. That's a very good question. So there is a selective process of including specific microRNAs, but then we also have a default mechanism. In all the conditions and stages where we looked at the microRNome of extracellular vesicles from different cell types, from different senescence, non-senescent cells, we see that about 50% of the microRNAs, they just stay the same level. So they seem to go there by default. They seem not to be really selected for to go into the extracellular vesicles, reflect more or less what you would find in terms of levels in the cytoplasm. But then about a fourth of the microRNAs that we find in the vesicles, they are de-riched in the cytoplasm, and another fourth or 25% is enriched in the extracellular vesicles as compared to cytoplasm. So there must be a specific mechanism. There is one study out that shows that there are some sequence-specific signals that lead to the inclusion of some microRNAs, but definitely there is a lot to discover in that regard. Can we manipulate that? We don't yet understand everything so well. We know some steps in the assembly and how the microRNAs get in there, so it is going to be you can manipulate that. And one thing, without knowing really the mechanism how you can manipulate, is transfect the cells with microRNAs, and they indeed go into the extracellular vesicles, as we have shown for the one specific microRNA, where we don't understand why this is happening. Why does it go into the... It goes in the endosomes probably, and then that's why and how it goes to the MVPs. But yeah, loads to do.
Thank you.
Cédric Delevoye.
Yes, here. Actually, I have two questions. One is related to more the biogenesis process within the MVB, since these senescent cells secrete more, as far as I understood. I was wondering whether you have a kind of new biogenesis, or it's just a kind of fusion process that would be kind of promoted. And a second question would be more related to the last part, that is, since you are able to collect extracellular vesicles from the fluids, I was wondering whether you can get numbers. I mean, because this is a real challenge now to try to understand whether it's a very dynamic, important process in vivo. So can you have numbers per cells, exosome per cells, whatever?
Mm-hmm. So the first question on the was biogenesis of extracellular vesicles. We see that the whole ER and all the secretory pathways are upregulated in senescent cells. That is something that is known. We haven't specifically looked at if the pathway to generate EVs would be up in senescent cells, but that's a very good question. I'll go right back to the lab and look at our expression data there. And the second question was on the quantitation of vesicles in the skin ex vivo. Yeah, so far we didn't do that yet because we had nothing to compare to. That was healthy individuals from a study that Your Name Research did, and where we were able to get some samples. So we cannot yet really compare, young versus old. This is something that we are planning at the moment. In terms of numbers compared to the in vitro situation, we are in a very similar range, and with about the tiny amounts that we get out of the perfusion system of about 100 microliters, we are able to isolate RNA out of that, and we are able to isolate the vesicles similar to, actually even better than from conditioned media. So, yeah, we are in the beginning of quantitating that and having the comparison, hopefully in a couple of months, which then at the end will be a couple of years.
Thank you very much, Johannes.